Hi guys, this is the lecture on meiosis, and before I begin I just want to make sure that you have a good foundation, that you've actually taken a look at the lecture on mitosis, because you're not really going to have a firm foundation and understanding of meiosis until you understand what's going on in mitosis. So please take a look at that before you take a look at this lecture, but if you have already, let's just keep going. Before I begin with the actual phases of meiosis and explaining how the chromosomes are broken down, I want to explain a little bit more about what a chromosome is, because now we're going to be getting into genetics, and we're going to be talking about sister chromatids. We're also going to be talking about chromosomes and referring to them as paired sister chromatids or single strands of DNA, and that can get really confusing. So I'd like to kind of go over the general form of a chromosome so that you understand and can visualize it when we talk about it. But basically, if this is our chromosome, okay, we actually have two strands of DNA here. There's one strand here, and then there's another strand there. And what's holding these two strands together is what we call a centromere. And a centromere is going to literally, it's kind of like a magnetic button. It keeps these two strands together. Now, coming off of the centromere, and we're going to be talking about this a little bit more through meiosis, is what we call a kinetochore. And I'll write this up here. But, and you might have seen this in your book already too, but kinetochore. Okay, and a kinetochore basically is these little kind of hairy looking things that come off the side of the centromere, and this is actually what's going to attach to spindle fibers. That's going to come in. So this is one giant spindle fiber. It's going to come in, attach to it. Okay, and that's actually what's going to attach to the spindle fibers, allowing the spindle fibers to basically rip that chromosome in half and separating that centromere. So literally what happens both in mitosis and meiosis is when those spindle fibers come in, they're actually going to attach not to the centromere, so to speak, but actually to little microtubules that extend from the centromere, and we call that kinetochores. And then once they've attached to that, the spindle is actually going to guide them and pull them apart and guide them to opposite ends of the cell. And if we separate this chromosome, we are only going to get two sister chromatids out of it. Now, during meiosis, there's going to be two divisions of this. So you're going to see whole chromosomes like this, a full chromosome, getting pulled to one side, kind of lassoing and being pulled to one side. And you're also going to be seeing what I just showed you, where a full chromosome is actually going to be split or pulled apart into two sister chromatids. Once that happens, sometimes these sister chromatids, and they kind of look like this, they still have a little centromere in them. Sometimes they are also referred to as chromosomes. As confusing as that sounds, you just kind of have to know what part of meiosis or what phase of meiosis you're talking about. Are we in meiosis 2 or are we in meiosis 1? If we're in meiosis 1, we're talking about the full chromosome. If we're in meiosis 2, we're probably, especially at the end of meiosis 2, we're probably talking about just a single strand of DNA or a sister chromatid, which sometimes can be still referred to as just a single chromosome. So let me just get this out of the way. Oops. And let's just bring this up. Hopefully you guys still have this from a while ago. I'm just going to make this a little bit bigger. But as we talked about in mitosis, a cell, just a general cell, is going to go through interphase. And interphase is basically just the general life cycle of the cell. The cell grows, it repairs itself, it just kind of lives, metabolizes, and then all of a sudden there's some kind of response. It needs to respond to a need. Maybe it needs to, help by adding more mass to an organism in order, to divide. Maybe it just needs to divide in order to replace a dead or damaged cell. Who knows? But that need arises, and it's going to answer that. So the big question here that you're probably wondering is, well, where do we get these gametes from? Where do we actually get sex cells from? Because in meiosis, we're not dealing with taking DNA and completely replicating it and then just dividing it completely in half. We're actually dealing with giving a daughter cell, which is at the end of this whole process, a different set or different combination of genes and DNA or genetic information than the parent cell started out with. And that's how we get all these variations in life. That's how you look different from your sister or from your parents, with the exception of twins, however, identical twins. However, there are some slight differences even there. But let's get back to this. So the cell, just a general cell, is going to go through interphase, and then a need is going to arise. So the cell is going to go through interphase, and then all of a sudden it needs to separate. So basically, meiosis 1, which you should see up here, okay, kind of looks a lot like mitosis, okay? A cell, just a general cell, a somatic cell, and hopefully you've gotten your notes off of the homework website, but it's going to start out with 46, and I'm just going to write chromosomes like that, okay? And it's going to replicate its chromosomes. So you can do the math. What's 46 times 2? I believe it's 92. So regardless, you're going to have double the amount of chromosomes and genetic material in the cell, okay? So you're going to have 2. So in meiosis 1, you're basically going to get, at least in metaphase, let me draw just a picture of, this would be metaphase, okay? You're essentially going to have all these full chromosomes lining up on the metaphase plate. Because remember, you now have 2 sets of them. And basically what's going to happen, just like I explained before, these fibers are going to come in, attached to the kinetophores on each one, and instead of what we see normally in mitosis, for instance, instead of separating the full chromosome and splitting it in half, you're actually just going to be pulling a chromosome, a full chromosome, over to one side or the other. Now in your notes it also talks about this is where crossing over occurs. So what's crossing over? Well, these pairs right here actually get really close to each other. In fact, they kind of look like this. They actually cross over, and you can kind of see that here. And basically what happens is at those points, and I wish I could show you in color, but we'll do that in school. Let's say that this is mom's chromosome and this is dad's original chromosome, okay? They're actually going to switch parts, okay? So maybe this part actually jumps onto this part, okay? And maybe this part switches with this part, okay? That's crossing over. So now you're getting a chromosome that might have one part from mom and maybe the rest of this is from dad. Who knows? Okay, but that's crossing over. So that happens on average about 3 times for every chromosome, and that's on average. So it's like life is definitely trying to make sure that there's a variation in genetic information there. So at the end of meiosis 1, you're still going to have 46 chromosomes here and 46 chromosomes here, okay? Plus we've had some crossing over. Plus, we have no idea how these have lined up on the metaphase plate during meiosis 1. So that's where we get our independent assortment from. These have totally, after crossing over, some of these might be moms, some of these might be dads, this might be a mom, this might be a dad, who knows? They completely randomly line up on that metaphase plate and then they're going to be divided, okay? So, now, and if you're confused about the math, 46 and 46 should equal approximately 92 or double that, okay? So now these cells, just like in mitosis, okay, nuclear envelopes are going to begin to form around the chromosomes. And remember, we still have these full chromosomes. We're not dealing with sister chromatids just yet, but we still have full chromosomes. Those chromosomes are going to decondense back into chromatin. Obviously during telophase, the spindle fibers will have broken down, okay, prior to cytokinesis, although it happens pretty quick. So spindle fiber breaks down, or the whole spindle breaks down, and then you have DNA decondensing into chromatin. Once that happens, instead of the cell going right into interphase like it normally would in mitosis, it goes right into prophase 2, okay, and we call it prophase 2 because now, and you can see this nice dotted line here. Now we're going to be going into meiosis 2. okay? And this is going to happen simultaneously to both cells, okay? So this is a pretty quick process, okay? So I'm just going to bring this up real quick, make it a little bit bigger, okay? All right, so this is meiosis 2 now, okay? So this is going to be prophase 2, metaphase 2, anaphase 2, and you guessed it, telophase and then cytokinesis. Okay, telophase 2 and then cytokinesis. So now we're dealing with 46 chromosomes in each. Okay, and the end result, obviously, to make gametes, we need to have half that number. We need to have 23 chromosomes in each of those 4 cells. So we're literally going to try to split this in half on both sides, and we're going to get 1, 2, 3, and finally 4 daughter cells, hopefully, okay? So during prophase 2, it's exactly like mitosis. The nuclear envelope is going to begin to break down, releasing those chromosomes, and remember, at this point, we still have those full chromosomes, okay? So they're going to look completely like an X still, okay? So that's going to happen. You're going to get your centromeres that are, or your centrosomes, excuse me, that are going to float to opposite ends of the cell and start producing those microtubules that are going to create that mitotic spindle, okay? And those chromosomes are going to try to kind of make their way toward that metaphase plate. And at that point, now you've gotten down to metaphase. So again, instead of dealing like we did up here, instead of dealing with 2 lines of full chromosomes, you're actually only going to deal with what you're probably more familiar with, 1 line of chromosomes, kind of like this, okay? And again, here's your spindle fibers. The middle is going to be attached, obviously, to the kinetochore. That's those microtubules, those special microtubules that kind of jet out from the centromeres of the chromosome. And then same thing over here is happening, so not to confuse anybody. Okay, this is where you start seeing that chromosome get totally split in half. Okay, so now, oh, I'm sorry, this is metaphase. I'm getting ahead of myself. Okay, so chromosomes align on the metaphase plate. Okay, so now what's going to happen? Well, now you're going to be pulling apart those sister chromatids. Okay, same thing's going to happen over here. Okay, all right, so now real quick, another reminder. These are called sister chromatids, again, because they are one strand of a full chromosome. But sometimes at this point, we can call them chromosomes. That's where, this is what I was talking about before, where you just kind of need to know where you're at in the process. So if I refer to these as chromosomes, but you know that we're in meiosis 2 towards the end, so, you know, they've already been separated and everything, then you'll know kind of what we're talking about as far as, okay, this is a sister chromatid, but we're referring to them as chromosomes. So they get split apart during telophase. So this is telophase 2. Same thing happens. Okay, so we've got, that's kind of what they look like. Here's your little centromere. These are single chromosomes. Okay, same thing over here. And remember, these single chromosomes, or sister chromatids, have different parts, genes from mom and genes from dad, okay, so to help with that variation. So now you're going to have your nuclear envelope beginning to reform around there. These are going to return back to a chromatin state, so they're going to decondense again. Your spindle fiber is going to break down, okay, so same thing that you are used to seeing. And then you're also going to see, again, that cleavage furrow begin to form right at the end of telophase 2. Same thing that happens after telophase 1. And that's usually a big sign that we're basically right in cytokinesis. So cytokinesis, again, is going to divide that cell in half, and in doing so, each half is going to kind of pull away from itself, making this center smaller and smaller until it eventually just pinches off in the middle, and you get 2 cells with 23 chromosomes each. So there's 23 there, 23 there. There'll be 23 there, and 23 there. Now, in males, these will all become sperm cells, obviously, but in females, only one of these will really become the egg. The other three will kind of be reverted back to a regular cell, but really, because females don't produce as many sperm or as many sex gametes as men produce, obviously sperm, they don't need all four. They just need the one. Okay, so if you have any questions about this, we'll talk about it in class. All right, thanks. Oh, one other thing I forgot to mention. I want to talk to you guys about the difference between haploid cells and diploid cells. A haploid cell would have a set of chromosomes from mom and a full set of chromosomes from dad. So you have two sets. Okay, and usually when we write diploid cells, so this would be an example of a diploid cell because 23 chromosomes come from mom and 23 chromosomes come from dad. So we got basically two full sets there of chromosomes. If I go back down here, basically these sex gametes, because they only have 23 chromosomes in each, these are what we call haploid cells, okay? And haploid cells are represented by just a little n because they only have a single set of chromosomes, okay, for that individual. And one other thing that you should know is that we've been talking a lot about human sets of chromosomes, where humans have a full 46 chromosome count in their somatic cells, and then 23 in their sex cells or sex gametes. But not every organism is like that, okay? Some organisms have, say, 3 sex chromosomes, okay? Some have 1 sex chromosome. We have 2 sex chromosomes. That's male, and then obviously this is female, okay? So just a quick note on that. We'll cover some more of that in class. Thanks, bye.